Welcome back to this next video in which we are discussing multiple sclerosis. Okay, so we're currently in the process of discussing the pathogenesis of multiple sclerosis, and the point in the story that we've got up to so far is that we have our individual here who is unfortunately going to develop multiple sclerosis. And we've discussed that in order for him to develop multiple sclerosis, he's going to have to develop an adaptive immune system that is capable of attacking a protein of the central nervous system, myelin. And we have called this protein that's going to be attacked the central nervous system myelin protein, just to give it a name, because we don't actually know its identity at our current level of knowledge. Now, the arm of the adaptive immune system that is specifically going to attack this protein in multiple sclerosis is going to be the extracellular arm involving CD4 positive T cells and B cells. So this individual is going to develop CD4 positive T cells directed against a peptide fragment of this CNS myelin protein, and he's also going to develop B cells that are directed against an epitope of this central nervous system myelin protein. Now, at the moment, the point in the story that we've got up to so far is that these cells are going to be in the naive state. Both the CD4 positive T cells and the B cells, they're going to be in the naive state. They've never been activated so far. And it might be the case that they sit in this naive state for many, many years before ever becoming activated. And whilst they're in the naive state, this individual is not actually going to have the disease multiple sclerosis. Only once these cells have actually been activated is this individual going to actually start encountering problems and potentially develop multiple sclerosis. So that's an important point to understand, that you develop the naive cells, and then there can be a large lag period, a large gap before this adaptive immune response involving these cells is actually going to be initiated, and only once it's been initiated will you actually get the disease. So, the question that we now turn on to is, how does this adaptive immune response here, this autoreactive adaptive immune response, actually get initiated? Now, you might think, well, isn't this the most obvious question in the world? If you've got these autoreactive immune cells here directed against this protein, and you've got a central nervous system, a brain and spinal cord that's full of this protein, then surely they're going to be activated by the presence of this protein. Surely this protein is going to activate this autoreactive adaptive immune response, and then the disease is going to begin. Well, the answer is we don't think that that's actually how this adaptive immune response is going to be initiated, and let me try and justify basically at this point why that actually is, but you'll understand more later on. But briefly at the moment, in order to activate the B cells, you need to have got activated CD4 positive T cells. I've said that already, that B cells cannot be activated without CD4 positive T cells that are directed against a peptide fragment of the same protein that the B cells are directed against an epitope of. So how do we activate CD4 positive T cells then? Well, in order to activate CD4 positive T cells against this protein, you would have to get a phenomenon called antigen presentation occurring. You would have to get this peptide fragment from the CNS myelin protein being presented in a very special way to the CD4 positive T cells, and it's that thing that we don't think is going to happen. We don't think that this protein is actually going to be antigen presented to the CD4 positive T cells normally, and therefore we don't think that this protein is actually going to end up being the thing that activates the CD4 positive T cells, and therefore the thing that activates the entire adaptive immune response. So that's a very important point to understand, that even once you've developed the ability to get multiple sclerosis, you've developed the adaptive immune system that could lead to getting multiple sclerosis, it's not actually a sure thing that you will ever actually get this adaptive immune response being initiated and therefore end up with the actual disease, because the protein we don't think is what actually activates this adaptive immune response. So now there's a mystery. How then does this adaptive immune response get activated? Well, instead we think it's going to be activated by an infection. You're going to get an infection potentially somewhere completely different in the body. In fact, almost certainly somewhere completely different in the body, because infections of the brain and spinal cord are actually very rare. So you'll get an infection somewhere else in the body, potentially in your respiratory tract or in your gastrointestinal tract or in your urinary tract, the common places that humans get infections. And then this pathogen is going to end up being able to activate this adaptive immune response that could then attack the CNS myelin protein, and that's going to be how it occurs. So there's a brief answer to that. Let's now explore this in much more detail. So let's come up here. So the baddie here then is going to be some pathogen, and I'm afraid here is where there's another hole in the explanation that I'm going to give you. We don't know what this pathogen is. So how am I going to represent this pathogen? I'm just going to draw a red circle here. So this is going to represent this pathogen that is going to infect you and trigger this autoreactive adaptive immune response. So this is some sort of infection. So this pathogen, what actually is it? We don't know is the answer. We don't know which part of your body it's actually infecting, but it's going to infect some part of your body, and it is some form of pathogen. We think that it's probably a virus. There are lots of candidates for what this pathogen is, and most of them are viruses, but I will put a question mark there. We think it probably is a virus, but we're not absolutely sure. What we can say is that our best current theory of how this adaptive immune response is going to be initiated is that some pathogen is going to infect some part of your body, potentially very far away from your brain and spinal cord, and that's going to trigger an adaptive immune response. And one of the adaptive immune responses that's going to be initiated is this autoreactive one. So how is this pathogen actually going to trigger the autoreactive adaptive immune response? Well, this pathogen is going to have a protein, which I will draw rather crudely as a great big box here. So here is one of the proteins of this pathogen, and we'll refer to this protein as an antigen. So an antigen is anything that can have an adaptive immune response initiated against it. So pathogens have loads of antigens, and most of them are proteins. So this is going to be one of the antigens of this pathogen. So it's a protein that this pathogen has associated with it, and it's one of the targets that when we're trying to defeat this pathogen, we're going to initiate an adaptive immune response against this antigen. So here is the absolutely crucial bit to understand. What if this pathogen has a protein that has the same peptide fragment in it as the peptide fragment that was present in the CNS myelin protein that the CD4 positive T cells were directed against? What if also it has the same epitope in it as the CNS myelin protein epitope that the B cells were directed against? What if, basically, this protein can also end up initiating this adaptive immune response that could attack the CNS myelin protein? That's what we believe happens. Now, this phenomenon where an antigen on a pathogen is very, very similar to one of your own proteins, and you end up attacking the protein on the pathogen only for the adaptive immune response to be able to attack your own protein. This is believed to be the way that a huge number of autoimmune diseases are initiated. So this theory, this understanding here, it doesn't just apply to multiple sclerosis. If you go on to study loads of other autoimmune diseases, you'll see the same theory is there in many of the theories of them. Okay, so what is the name then for this idea that a pathogen could have an antigen that is too similar to one of your own proteins? This is the idea of cross-reactivity. So this is the fancy name for this concept that I'm teaching you about now. Cross-reactivity. It means that the adaptive immune response, or one of the adaptive immune responses that we're going to initiate against this antigen of this pathogen, can also cross-react with a self-protein. So a very, very important concept, cross-reactivity, for understanding autoimmune diseases. Now, an important thing to say about when we initiate adaptive immune responses against pathogens is that This pathogen will have a huge number of proteins and therefore a huge number of potential antigens that we can initiate adaptive immune responses against. Each of these antigens can be chopped up in a huge number of different ways, i.e. it has a huge number of peptide fragments associated with it. So there is the possibility of initiating a huge number of adaptive immune responses against a single pathogen. I.e., we might have loads of different clones of CD4 positive T cells that are directed against different peptide fragments that are present in the different antigens of this pathogen. And we might have loads of different B cell clones that are directed against the different epitopes of the proteins of this pathogen. So important to understand is that a pathogen can initiate loads of different adaptive immune responses, not just one. We are, of course, interested in the fact that this pathogen, because one of its antigens might have the same peptide fragment that is within the CNS myelin protein and the same epitope that's within the CNS myelin protein, that we have autoreactive CD4 positive T cells and B cells directed against respectively, that we could end up initiating that specific adaptive immune response, and then it could then go on and attack the CNS myelin protein. Now, before we actually talk about how the adaptive immune response is initiated, I firstly want to talk through inflammation with you. So we are going to get infected by this pathogen that we do not know exactly what it is, but there are lots of candidates for it, and if you want you can go and look up what the candidate pathogens to trigger multiple sclerosis are, but we haven't got one that we're absolutely sure of yet. We're going to be infected by this, and what we've said so far is that the way that it's going to actually end up initiating the adaptive immune response is this cross-reactivity principle. It's going to have an antigen that cross-reacts, an antigen that can initiate an adaptive immune response that cross-reacts with the CNS myelin protein. But I firstly actually want to talk through what's actually going to happen when you get infected with this pathogen. I want to talk about the inflammatory response, i.e. the innate immune response, and then how the adaptive immune response is initiated. I want to go through it in the proper order, and the reason I want to spend some time talking about inflammation is, of course, that inflammation is going to come up later when we talk about how the white matter lesions are actually going to form in the central nervous system. So I want to make sure that everyone's actually familiar with how it's supposed to work, how when you get a pathogen infection you first get inflammation and then you get the adaptive immune response, so that we can totally understand the difference between that and an autoimmune response where it's the adaptive immune response going first and then the inflammatory response. So let me just talk you through what's going to happen when this pathogen infects. So, we have this pathogen here. It's now going to infect some tissue, and again, we don't know which tissue it's actually going to be infecting. As I say, the most common tissues for humans to get infected are, of course, the respiratory tract. That's what colds and the flu is. The gastrointestinal tract, that's what gastroenteritis is. And the urinary tract, that's what a urinary tract infection is. Also, the skin is a common site where humans can become infected. So it could be infecting any one of these tissues, but we don't know the answer as to exactly which one it is, because we don't know, of course, what the pathogen is. So this pathogen is going to infect some tissue, and it's going to start replicating. Either if it's a extracellular pathogen, it will replicate outside of the cells, or if it's an intracellular pathogen, such as a virus, it will have to go inside cells to replicate. And you might be thinking, wait a second, isn't there a problem here? If you're clued on, you might be thinking, isn't there going to be a problem here? If we think that the likely contender for this pathogen is a virus, that's an intracellular pathogen. So surely you would not initiate an extracellular adaptive immune response to a virus. And the immune response that we're hoping to get initiated here is we're not hoping, but which we're trying to understand how could be initiated, is an extracellular adaptive immune response. Surely there's something wrong there. But you also have to remember that viruses, they're not always inside of cells. They will sometimes be in the extracellular fluid. Just to draw a simple picture of this, so let's have some body cells here. So here are some human cells. And then let's have a virus, and the virus can just be this red blob here. So, when a virus infects your tissue, what happens is it's initially in the extracellular fluid, then it will go inside a cell, it will use the cell to make loads more viruses, and those viruses will then escape from this cell and go on to infect other cells. The point is that the virus spends some time inside the cells and sometimes outside of the cells. In between infecting cells, it is in the extracellular fluid. So actually, when you have a viral infection, you usually get both the extracellular and the intracellular arms of the adaptive immune response being initiated against that virus. The extracellular arm of the adaptive immune response can attack the virus whilst it's in between cells, whilst it's in the extracellular fluid, and the intracellular arm of the adaptive immune response can attack the virus whilst it's actually inside the cells. The CD8 positive T cells, that is, they can actually cause virally infected cells to kill themselves and kill all the viruses that are inside them, therefore. So, if you were wondering that question, there's the explanation. Viruses don't just have the intracellular adaptive immune response initiated against them. They have intracellular and extracellular arms of the adaptive immune response initiated against them. Okay, but let's take a step back. So, we have this infection by this pathogen, and it's either going to be an extracellular pathogen or an intracellular pathogen. What is actually going to happen initially? Well, the first part of the immune system that's going to attack this infection is not going to be the adaptive immune response, it's going to be the innate immune response. Inflammation is going to occur. Remember, we talked about the innate immune response. You get proteins coming in from the blood. You get the two basic types of white blood cells, neutrophils and macrophages, coming into the tissue as well. And let me just talk you through how this is going to occur. So, when you have a pathogen in tissue, and I'm just going to draw my pathogen here. So here is the pathogen, and it's in your tissue. How do you actually realise that there is a pathogen present within a piece of tissue and then initiate an immune response against it? How do you initiate the inflammatory response is the question. Well, the answer is that there are special cells in every tissue of your body that are standing guard looking for pathogens. And these cells are known as sentinel cells. So sentinel cells refers to guard cells. So they stand on guard looking for pathogens. Now, in most tissues around the body, the two major types of sentinel cells are resident macrophages and dendritic cells. It is slightly different in certain tissues of the body, in particular in the brain it is different. There's a cell called the microglial cell. However, this initial infection by the pathogen that's going to initiate this adaptive immune response that's going to attack the CNS myelin protein, it almost certainly is probably not in the central nervous system. It's going to be in some other tissue, because infections of the central nervous system are incredibly rare and incredibly dangerous. So it's going to be far, far away, so it's most likely going to be the normal types of sentinel cells that are going to be present in this tissue. So, let me draw these normal types of sentinel cells. So we'll put one here, and this is the dendritic cell, which is why it's got this sort of star shape here. So I'll put its title down here. So it is an example of a dendritic cell. Now, these processes coming off its cell body, these are of course the dendrites, and it has absolutely nothing to do with nerve cells. Dendrites are just referring to these processes coming off the cell body. They're nothing to do with nerve cells. This cell is not capable of conducting action potentials. Quite simply, it does not have the appropriate voltage-gated ion channels in its cell membrane that you need to conduct action potentials. So don't think that this has anything to do with neurons. It's got dendrites because it wants to extend these dendrites into as much tissue as possible, because it is looking out for signs of pathogens. So these dendrites are effectively its arms. They're the way that it can look for pathogens in a huge amount of tissue. Okay, so that's the purpose of these dendrites, to increase the surface area or increase the volume of tissue that this dendritic cell can stand guard over. Now, for short, we like to abbreviate dendritic cell down to DC. Okay, so that's one example of a sentinel cell that's present in loads of tissues around the body, so all over the tissue, all over the body, in the gastrointestinal tract, the respiratory tract, the urinary tract, you will find dendritic cells standing guard. The other major type of sentinel cell that's found in a huge number of tissues, and I'll change colour for this, we'll go to blue, is the resident macrophage. Now, macrophages are one of the major types of cell that's going to be brought in as part of the inflammatory response. However, you also have resident macrophages in a huge number of tissues within the body. So this is a resident macrophage, and these ones are here permanently. So macrophages, two big roles. They're dotted around tissues all the time, so loads and loads of tissues in your body have these resident macrophages dotted around, and they're performing a sentinel role. But then also, if you actually do initiate an inflammatory response at a certain piece of tissue, then you bring in loads more macrophages, so they're one of the major cell types of the inflammatory response as well. So two big functions that macrophages have here. So, macrophages, as I previously said, they're a great big blob of a cell like so, and here's its nucleus. So, these are the two major types then of sentinel cells, the dendritic cells that look this star shape and the resident macrophages, and these will be dotted around a huge number of tissues, and their job is to stand guard and look out for signs of pathogens, and if they find signs of pathogens, their job is then to set off the alarm and initiate the inflammatory response. So, the next question is, how do they actually detect pathogens? Well, I've already given you this answer in previous videos. The way that they do it is by looking for molecules associated with pathogens that no human cell in its right mind would ever create. So by looking for molecules that no human cell would ever make or use, you can be sure that you're dealing with a pathogen. If you see these molecules, you know that something is wrong. You know that there is some other form of life in the extracellular fluid that is not a human cell and that should not be there. Now, there are a huge number of different molecules that only non-human cells would ever use. However, to simplify everything down, we have a beautiful term, an umbrella term, that encompasses all of them. And this term is PAMPs, and it stands for Pathogen-Associated Molecular Patterns. So the first P is for pathogen, the A is then for associated, the M is for molecular, and then the final P is for patterns. So pathogen-associated molecular patterns, PAMPs for short. So this is a great big umbrella term that covers all of these molecules that no human cell in its right mind would ever make or use, and which are a giveaway sign that there is a pathogen present within your tissue. So, the sentinel cells, the resident macrophages and the dendritic cells, they have on their surface receptors for pathogen-associated molecular patterns, and if these receptors get activated, then the sentinel cells know that a pathogen is present. So, our pathogen here that is going to initiate the autoreactive adaptive immune response of multiple sclerosis, it's going to also have pathogen-associated molecular patterns associated with it, and these are going to lead to the activation of these sentinel cells. So the sentinel cells will become activated in the tissue that this pathogen is infecting, and I'll say it again, we don't know which tissue it actually is because we don't know what the pathogen is. What will then happen is the initiation of the inflammatory response initially, and pay close attention to this. If your immunology is not very strong, this is the normal type of immune response, basically. When you have a pathogen infecting tissue, this is what happens normally. You get the inflammatory response occurring first, and then much later you get the adaptive immune response occurring. And I really wanted to include this discussion so that you understand the difference between the normal stages of the immune response and what's going to happen in our autoimmune response in the case of multiple sclerosis, so that you can compare and contrast. Okay, so what happens normally in the presence of a pathogen when sentinel cells find a pathogen is that they initiate the inflammatory response, and the way that they do this is by releasing a whole bunch of alarm molecules to tell the microvasculature blood vessels to initiate the inflammatory response, to start recruiting in troops from the bloodstream. And I should just give that motivation for the inflammatory response. We have a pathogen present within this tissue. How are we going to eradicate this pathogen? Well, all of the troops to eradicate the pathogen are within the blood. And I should say that macrophages and dendritic cells, these sentinel cells that you have, they're pretty scarce. They're dotted around the place. They can fight the pathogen themselves. They can actually take part in phagocytosis. They can engulf the pathogen and break it down. However, there's not enough of them, so they need to call for help at the same time. And this is the major thing they do. They call for help to bring in loads more troops to try and eradicate the pathogen. And the troops are in the bloodstream, so fundamentally the response that we need is a response of the tiny little blood vessels in that tissue. We need them to start recruiting proteins from the bloodstream and recruiting white blood cells from the protein. We need all of these troops to come in and help us eradicate this pathogen. So that's the motivation for what we're trying to achieve here. So they are going to release pro-inflammatory mediators then, the alarm signals that are intended to set off the inflammatory response. Now, the two major pro-inflammatory mediators, and I'll firstly just put the title pro-inflammatory mediators here. So they're going to release pro-inflammatory mediators. So these are molecules, little signaling molecules, that are going to trigger the inflammatory response. Now, there are absolutely loads of these. If you want, you can go and learn lists and lists of pro-inflammatory mediators. However, if you don't want to learn a great big list of pro-inflammatory mediators, there are two that are absolutely essential to know about, that you must know about. The two most important pro-inflammatory mediators, and these are interleukin-1 and tumor necrosis factor alpha. So we will just put these two down because these are the two most powerful ones, the two most important ones. And this goes for all initiation of the inflammatory response. Anywhere, when you're studying the inflammatory response, these two mediators are the most important ones to know about. So interleukin-1, abbreviated down to IL-1, and then the other one is TNF-alpha, which stands for tumor, that's the T, necrosis, that's the N, and then the F is for factor, and then it's the tumor necrosis factor alpha. There are other tumor necrosis factors as well, but tumor necrosis factor alpha is easily the most important one. Okay. So, our sentinel cells have detected the sign of this pathogen that is infecting our tissue, and they don't like this, so they're setting off the alarm, and they're releasing these pro-inflammatory mediators into the extracellular fluid that are going to have an effect on the microvasculature. I've said that there are absolutely loads of these, but if you're just going to learn two of them that must stay in your mind forever, their names are interleukin-1 and tumor necrosis factor alpha, and both of these are protein mediators that the sentinel cells have synthesized. Now, another little piece of terminology that people often get confused about is the term cytokine. You will often hear interleukin-1 and tumor necrosis factor alpha referred to as pro-inflammatory cytokines, and people often get confused about what this word actually means. What's the difference between a cytokine and a mediator? Well, this term pro-inflammatory mediators is broader than the term pro-inflammatory cytokines. A pro-inflammatory mediator is anything that can be released by cells to cause inflammation. A pro-inflammatory cytokine has a slightly narrower definition. This is a molecule released by cells of the immune system, and these sentinel cells, the macrophages and the dendritic cells, they are certainly cells of the immune system, so tick that box. They're molecules released by cells of the immune system that go and affect the behavior of another cell. Okay, that's obvious. And they also have to be proteins. So let me say that again. Pro-inflammatory cytokines are proteins released by cells of the immune system which will go and act on other cells, not necessarily cells of the immune system. Indeed, these are going to go and act on endothelial cells of the microvasculature, and they will change the behavior of those other cells. Okay, so that's why mediator is broader than cytokine, because mediator doesn't put the requirement that it needs to be a protein, whereas cytokines are proteins. Interleukin-1 and TNF-alpha are both proteins, so you can refer to them as pro-inflammatory cytokines if you wish. Okay, so what's going to happen next? The pro-inflammatory mediators, interleukin-1 and tumor necrosis factor alpha, are going to go to the microvasculature blood vessels. So they're going to go and act on the tiny little blood vessels within the tissue. So the tiny little arterioles, the tiny little capillaries, and the tiny little venules, which we can overall call the microvasculature. They'll cause changes in those blood vessel wall state, and the blood vessels will now start allowing proteins from the bloodstream to leave the bloodstream and go into the extracellular fluid. And of course, when the proteins move, they have to move with a fluid as well. So we call that fluid that accumulates in the area an inflammatory exudate. So you're going to get inflammation, and the features of inflammation are you're going to get something called an exudate forming. And I'll say again what I mean by an inflammatory exudate. So when the inflammatory response occurs in a portion of tissue, the little blood vessels are going to start allowing these proteins that have been made in the liver and which are very good at attacking certain forms of pathogens, but they are quite broad. They can attack a huge number of different pathogens. But they're not specific, of course, the proteins that are made by the liver. They attack a huge number of different pathogens, but they do it in exactly the same way. So they're not specific to pathogens at all. So we want these proteins to come into the site where the pathogen is actually present, and when they come in, they are going to bring water with them, fluid from the bloodstream, and that mixture of fluid full of proteins that's going to accumulate at the site, and this is one of the reasons that inflamed areas swell because of this inflammatory exudate, is called an exudate. So when you've got a fluid that is coming out of the bloodstream and is full of proteins, we refer to that as an exudate. And when it's because of inflammation that it's being formed, we call that an inflammatory exudate. Okay, so one of the features of inflammation is that you're going to get an inflammatory exudate with all those proteins being formed at the tissue, and that's going to help, hopefully, to clear the infection. And the other big thing that's going to happen is, of course, you're going to bring in white blood cells, which are abbreviated down to WBCs, and the two major types of white blood cells that you're going to bring in are the neutrophil, which I'm abbreviating down to N and then the Greek letter phi, and macrophages, which are abbreviated down to N-phi. Now, just to draw some pictures of these cells then, we'll go to another colour. Let's pick a exciting new colour. We'll have vivid purple. So let's draw a picture of a neutrophil, because we haven't done that so far in this video. So a neutrophil has a strange nucleus. So it's a nice round cell, as most cells are, and then its nucleus is very strange. It only has one nucleus, but its nucleus has a very strange shape. It has lobes, like so, and it's usually got about 3 lobes, like this, and those lobes are connected by little thin bits, little I don't know what to call it, little tubes. So the nucleus is multi-lobed with little connections, obviously, between the lobes. So this is the strange shape of a neutrophil nucleus. So this is what neutrophils look like. As I've said previously, they're quite a cheap cell of the immune system. They're a very basic cell. They don't live very long. They live for about a week in the blood, and you have millions, if not, you have billions, in fact, of them in your blood. It's usually between 2.5 and 7.5 billion neutrophils per litre of blood, and you've got about 5 litres of blood. So you've got absolutely billions of these neutrophils within your bloodstream at any one time. They don't live long. Your bone marrow is continuously producing more of them, and they're a very, very basic cell. They are not specific, they're not clever, they're not difficult to understand like the T cells and B cells that we spent so long discussing. They're a very basic cell, and the way that they can attack pathogens is that they can eat them effectively. They can engulf them inside and break them down, and that process is known as phagocytosis, and I will just draw a little picture of the basic idea of phagocytosis. So they can phagocytose pathogens. So the idea is this: if we have a pathogen here, what can happen is the neutrophil can come up here and it can try and engulf the pathogen, and the way that this occurs is that it invaginates its membrane. So if I just go back to the pink, and I'll draw this a little bit more in detail. So what I'm drawing here is a little section here zoomed in on effectively. So here is the pathogen, and you can see what's happening. It is invaginating its membrane inwards, and then eventually what will happen is this will completely pinch off. So it will go to this, and then it will go like this. So you'll have a little vesicle now, a phagosome as it's called, with the pathogen inside, and then what will happen is the neutrophil will break down the pathogen, or at least in principle it will have a very good go at breaking down the pathogen inside that phagosome. So just to put that key piece of terminology on here, so it's forming a phagosome here. And that process of engulfing pathogens and breaking them down, that's called phagocytosis, and that's the way that neutrophils can attack pathogens. Macrophages are a bigger, better version of neutrophils, effectively. So let's go to another colour. We'll go to aquamarine for a macrophage. So we've already drawn a picture of a macrophage. Macrophages are not circulating within the bloodstream. Instead, in the bloodstream, you have monocytes, which look kind of like this. They have a weird nucleus as well. It's often described as being kidney bean shape. So we'll just go down here. So this is a monocyte, and this is the precursor of macrophages that circulates within the bloodstream, and it's effectively a more compact version of the macrophage. Macrophages, as I previously said, are big blobby cells. You wouldn't want those big blobby cells circulating within the bloodstream, so instead you have a compacted down version of a macrophage called a monocyte circulating within the bloodstream, and then when this comes out of the bloodstream as a site of inflammation, it turns then into the great big blobby cell that we call a macrophage. And those macrophages, their major way of killing pathogens as well is via phagocytosis. Okay, so this is what the inflammatory response then is. Let's summarise what we have just seen. So, we are trying to understand the pathogenesis of multiple sclerosis. We've discussed that the way that you're going to get the initiation of this autoreactive adaptive immune response is by being infected with a pathogen that has an antigen that could initiate an adaptive immune response that would cross-react with the central nervous system myelin protein. Now, at the moment we haven't discussed this pathogen initiating the adaptive immune response yet. We're discussing how it would initiate an inflammatory response, the innate immune response. So it has infected some tissue. We don't know what tissue it actually is going to infect, but it's going to infect some tissue. And at that tissue, there will be sentinel cells, most likely resident macrophages and dendritic cells. These are the sentinel cells that are present in most tissues. And when these realize that there is a pathogen present within that tissue, and they'll do so by recognizing pathogen-associated molecular patterns on this pathogen, they will start releasing pro-inflammatory mediators, or indeed the two that we've named you could call pro-inflammatory cytokines. So the major two pro-inflammatory mediators are interleukin-1 and tumor necrosis factor alpha, so they'll start producing interleukin-1 and tumour necrosis factor alpha and secreting these into the extracellular fluid. These will go and act on the microvasculature blood vessels and will trigger a response in the microvasculature blood vessels. The microvasculature blood vessels will now start allowing proteins from the bloodstream along with fluid from the bloodstream to go into the extracellular fluid in that tissue. So you'll start forming an inflammatory exudate, and those proteins will hopefully start to attack the pathogen. You'll also bring in the two basic forms of white blood cells, neutrophils and macrophages, which are both phagocytes. So overall, at this site of inflammation, you're going to get loads of neutrophils and macrophages accumulating, loads of inflammatory exudate, and hopefully this will have a good go at killing the pathogen. And indeed, inflammation is very effective, even though this is very crude mechanisms compared to the adaptive immune response. It is effective in a huge number of cases at clearing the pathogen. However, what can now be initiated after the inflammatory response is the adaptive immune response. And if the inflammatory response alone is not sufficient to destroy the pathogen, then the adaptive immune response will work, hopefully. So, we now want to talk about how this pathogen is going to initiate the adaptive immune response. But I think we'll have a break here and we'll discuss the activation of the adaptive immune response in the next video. And of course, the adaptive immune response that we're very, very interested in is the fact that it will be able to initiate the same one that could attack the central nervous system myelin protein. So we'll see that in the next video.